In 2019 alone, antimicrobial resistance contributed to nearly 4.95 million deaths. It compromises medical treatments, increases mortality, and drives up healthcare costs. If left unchecked, it's projected to cause 10 million deaths and a $100 trillion economic loss by 2050. How does this happen? Bacteria evolve, mutating drug targets, pumping antibiotics out, or releasing enzymes that break them down. These mechanisms render even our strongest antibiotics useless. So we turned into one of the promising alternatives, antimicrobial peptides. AMPs are powerful natural molecules that can kill resistant bacteria. But finding effective ones isn't easy. Testing each AMP in the lab requires costly and time-consuming experiments due to millions of possible peptide combinations. To accelerate discovery, we used computational biology, testing over 600 combinations for classification and 2,000 for MIC prediction. We deployed the best-performing models in EPIC AMP to target E. coli, P. aeruginosa, S. aureus, and K. pneumoniae, all high-priority or critical pathogens. Our comprehensive approach with EPIC AMP yielded exceptional results. For peptide classification, our random forest model hit 96% accuracy, significantly beating the 91% literature benchmark and showing superior discrimination. In MIC prediction, we saw strong quantitative performance across key pathogens. E. coli achieved an R-squared of 0.7, S. aureus 0.68, and P. aeruginosa 0.68. Most notably, for K. pneumonia, our MLP model reached an R-squared of 0.74, consistently outperforming published regressors and underscoring EPIC AMP's robust potency prediction. Let's test our EPIC AMP platform, an AI-powered web tool for antimicrobial peptide prediction. EPIC AMP integrates classification and MIC regression models to assess the antimicrobial potential of peptide sequences targeting critical pathogens. Let's begin with the Usage Guide tab. It provides clear instructions, from sequence entry to model submission and downloading your report. Next, the Models Metrics and Sample Run tab. Here, we display our model's performance and SHAP global explainability with interpretations. Also, we added Sample Run for test. The About tab gives background on the tool's design and our team at Zewail City, as well as how the models were selected based on accuracy, robustness, and biological relevance. Now, let's return to the Prediction tab and run a full test. We'll upload a FASTA file containing candidate sequences for prediction. Then, we select all 4 bacteria, E. coli, P. aeruginosa, S. aureus, and K. pneumonia, for MIC estimation. We enter our email address to receive the report, and finally, click Submit. The system now processes the input using our trained machine learning models. This typically takes about 30 seconds, depending on sequence length and number of organisms selected. Once processing is complete, the classification result will appear here, AMP or non-AMP. Also, a full PDF report will appear and will be sent to your email. Let's check the email. As you can see, the report includes sequence, classification outcome, confidence, and predicted MIC values against the bacteria you target. This provides researchers with rapid insight, helping prioritize which peptides to test in the lab. With EPIC AMP, we bring machine learning into the hands of researchers, accelerating AMP discovery and helping combat antimicrobial resistance. Thank you for using EPIC AMP.